In the previous video, we have discussed that phosphorylation in the Calnexin at 563 serine position, or we can say that corresponds to MAPK signaling pathways that helps in the transport of TrKB to the cell surface. In this video, we will discuss. In this video, we will discuss that what will happen in the Calnexin depleted cells, why the TrKBs are not expressed in the Calnexin depleted cells on the cell surface. So the hypothesis that they used is that in the Calnexin depleted cells, the TrKBs are being directed to the lysosomes because of the unfolded structures. In order to validate that, they did this immunostaining of this LC3, which is the marker for lysosomes, and this KDL, M-cherry and TrKB. And in the wild type cells, they are able to see that there is a overlap. There is a almost overlap for all the threes, that is LC3, KDL, M-cherry and TrKB occurs, which is the indications of autophagosomes. So however, when they check for the calnexin depleted cells, you can see that it is not completely overlapping, but they are present at the close to the this TrKBs are present close to this KDL, M-cherry or this LC3, indicates that its autophagosome is been defected. And when they use these autophagy inhibitors, they can see the enhanced autophagosome in the calnexin positive cells or in the wild type cells, whereas in the case of these calnexin negative cells, they can still see the defect or impair autophagosome. And these things are being validated in this quantitative analysis, where we can see that in the calnexin negative cells, the calnexin negative cells, the amount of this autophagy, amount of this AP cells are higher even without these autophagy inhibitors. And when the autophagy inhibitors are used, they can see that a difference still exists. That is, the autophagy has been reduced. Autophagosomes are been less or weaken in the case of this calnexin negative cells compared to the case of wild type. And they have validated these results using fluorescence measurements at different positions, where we can see that in the absence of these autophagy inhibitors, there is almost overlap of TrKB and this lysosomes. However, for this calnexin depleted cells, we cannot see the overlap. Instead, we see that TrKBs are just close to the signals for this LC3, and when these inhibitors are used, autophagy inhibitors are used, we can see the complete overlap in the case of these wild type cells of this TrKB, LC3 and this KDL, M-cherry, that can be seen here. So this indicates that autophagosomes are increased when these autophagy inhibitors are used in the case of wild type cells. However, in the case of this calnexin depleted cells, this is not the actual scenario. Instead, they can see that some of this LC3s are just close to the TrKBs. And in order to validate this using immunostaining, they did this immunostaining of TrKB, LC3 and M-cherry, KDL, and they can see that some of the these TrKBs are lies in the peripheries of the LC3, whereas these KDLs and LC3s are seems to be overlapped. And in order to check whether there is an impact that occurs on the lysosomes. So this is about the lysosomes marker. Now they need to check whether the this is about the autophagosome. I need to check the lysosome effect also. In order to check the lysosome effect, they use this lysosome marker proteins, that is LAMP1. So when they use this LAMP1, they can see that in the wild type cells, the lysosomes are working normally. That is, they are acting on the almost all the TrKBs in the basal conditions. Whereas in the case of these Calnexin negative cells or Calnexin depleted cells, they can see some of these TrKBs that are left over. This further indicates that there is a weakening or impaired lysosome effects might be going on, and these things are being validated using the quantitative analysis, where they can see that the overlap of LAMP1 and TrKBs are higher in the Calnexin positive cells, but they are lower in Calnexin depleted cells. And when they check this LC3 at different time dependent along with this EGF injections, they can see that the LC3's amount goes down with time of these EGF injections. So this in the case of wild type cells and also in the case of this negative cells. However, in the wild type cells without any chemical inhibitors, they do not see any EGF LC3 formations even with EGF. When this chemical inhibitors are used or autophagy inhibitors are used, they can see the autophagosomes or LC3 signals. And however, for these Calnexin negative cells, they can see some uh, autophagosomes or LC3 signals even in the case of this without any autophagy inhibitors. And when the autophagy inhibitors are used, they can see the increased LC3 signals. However, when we check these high intensity signals, we can see that the signals of this LC3 without autophagy inhibitors and after autophagy inhibitors are almost similar. So this indicates that there is a impaired lysosomes in the Calnexin negative cells, and that does not increase further even when these autophagy inhibitors are used. However, in the wild type cells, when the autophagy inhibitors are used, they can see the increased autophagosomes, or in other way, the decreased autophagy, because autophagosome is the intermediate step for autophagy. And when they check for these TrKBs, they find that elevated levels of TrKBs when the chemical inhibitors are used. This indicates that the weakening autophagy that leads to the more accumulation of this TrKB. And same things that they have find out in the case of this Calnexin depleted cells with autophagy inhibitors. However, after a certain time, like after 4 hours, they can see the decrease in the TrKB levels. So this indicates that autophagism is the function of this EGF, as the EGF might stimulate the more autophagosomes, but weak EGF might stimulate more impair of the lysosomes in the Calnexin depleted cells. So, and however, when after sometimes in this case after 4 hours, they can see that the amount of this TrKBs goes down, indicates that the lysosomes might be functioning low. Lysosomes might be functioning weak, or the other things that they hypothesize that it could be one reason could be that there is a epigenetic changes or transcription changes occurs. Epigenetics or transcription changes occurs that leads to the low production of this TrKB. In order to validate this, they did their they check the mRNA levels. So before going to the mRNA levels, the quantitative analysis of this LC3 and EGF is shown in this panel G, where we can see that LC3 levels goes down in the Calnexin depleted cells when even in the case of this without chemical inhibitors and after chemical inhibitors. However, when these chemical inhibitors are introduced, they can see the elevated levels of this LC3 both in the case of this wild type cells and Calnexin depleted cells. But for the 4 hours, the difference between LC3 in the Calnexin positive cells and Calnexin depleted cells has been significant. So this could be because of in the wild type cells there is a lysosome functioning is already healthier, whereas in the case of this Calnexin depleted cells, lysosome functioning is already impaired. So the increase in LC3 levels in the Calnexin depleted cells is not much. So this that's what I have said, that EGF levels will also plays a role towards this lysosome impair. Next, this is the in this second figure of the panel H indicates about the TrKB levels, and in this case when no chemical inhibitors are used, all TrKB levels are same even in the case of both in the case of Calnexin depleted and Calnexin positive cells. However, for this when the autophagy inhibitors are used, they can see their statistical significance after 15 minutes and also after 4 hours, indicates that TrKB levels increased in the wild type cells. So from this they come to a point that it could be that mRNA expressions or transcription changes might have occurred, and in the case of these wild type cells there could be high production of TrKBs compared to the case of this Calnexin depleted cells. In order to validate this, they check the mRNA levels in the case the presence and absence of EGF. So when the chemical inhibitors are not used, they can see that TrKB mRNA's productions are higher in the wild type cells compared to the knockout cells, and when these chemical inhibitors are used, uh, the changes are still there but they are not statistically significant. However, when there is no autophagy inhibitors are used, they can see that the difference is statistically significant. So this indicates that there is a epigenetic transcription changes occurs in the case where uh, wild type cells are present compared to the case where Calnexin is depleted. And in order to validate this, so after this, so this has been validated that mRNA levels are low. So this lower in the level of TrKBs due to the epigenetic or transcription changes. Thereafter, they wanted to check whether it's only the lysosomes degradation occurs or the proteasomal degradations of this TrKBs proteins also occurs. So what they did is they prepare a cell system where they prepare a cell system where they knocked out they prepare a cell system where they use the markers for the proteasomal degradation, that is Mg132, and check the amount of this TrKB in the presence and absence of this Mg132. It is a marker for proteasomal degradations, and both in the case of this Mg132 present and Mg132 absent, they do not see a change in the TrKB. However, as a control they use ubiquitin, which would increase when these markers are used, as it will affect the proteasomal degradations. So this control shows that the Mg132 working, but there is no effect on TrKB. So the TrKB does not degrade through proteasomal degradations. So thereafter they decided to move further toward lysosomal degradations and decided to check whether it is the lip, lipofactions or at C3 site. Lipofactions at C3 site uh, affects this lysosomal degradations, or it's some other way that affects the lysosomal degradations. So in order to check that, they prepare this knockout with this. They prepare a cell lines with this CRE ATG5 uh, construct, and they validated their construct using this Western blot, where they can see that when this CRE is been when the CRE has been triggered, then they can see that ATG5 is not being produced. However, they can see the increased level of TrKB, and these things are being quantified in the panel M, where they can see that when the CRE5 is used, they can see the reduced level of this LC3, which is shown here, and it is statistically significant, and simultaneously they can see the increased level of this TrKB when the CRE is introduced. So this indicates that is the lipofation lipofactions at this LC3, lipofactions at LC3 or lipofactions of lysosome uh, has some contribution towards this TrKB degradations. So I hope you got some overview, as this is a lot of informations in this figures, and if you need to understand in detail about these experiments and their results, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.